Pulling up her slides. Our second speaker is Dr. Grace Chen. Grace is an assistant professor in the Department of Immunobiology and a member of the Cancer Immunology Research Program. She received her PhD at Harvard, where she worked on characterizing RNA modifications, her postdoctoral work at Stanford, and now I think her lab is really advancing an understanding of the intersection between the function and regulation of RNA, immunobiology, and beyond. So thank you.
Thank you, Charlie, for the introduction and for the opportunity to present some of our work that we're doing to understand the landscape of circular RNAs. So I chose this map that's a little bit older because you might see that the major continents are there, but there are key landforms like California and part of Australia that's missing. And so similarly, I think in RNA biology, there's still a lot to discover and uncover in this area. We typically think of RNAs as being spliced to yield consecutive exons in a linear fashion, typically with a 3' poly A tail and a 5' cap. And often the first step in RNA sequencing involves selection of all the RNAs with this poly A tail. And then we sequence them and we characterize them. And that actually means that we miss all the other RNAs that lack this poly A tail. And so technology has been a really strong promoter of biology and basic science discovery. And I think, even though we've had next generation sequencing for a while, it took quite some time in order to discover this whole new class of RNAs. So about 7 or 8 years ago, investigators took total RNA, depleted ribosomal RNA, and then sequenced everything that was remaining. And they found evidence of back splice junctions, where, for example, exon 3 is upstream of exon 2. And they found that this could only take place from templates of circular RNAs. And this really launched an entire new field where people are trying to understand what circular RNAs are doing and, first identify what they are. So this shows a phylogeny tree where species in red have been found to contain circular RNAs, and I've labeled the metazoans that also contain circular RNAs in gray. And actually, since 2014, essentially every eukaryote that's been looked at has encoded circular RNAs. So some of the individual circular RNAs that have been found in metazoans are labeled up here, and people have shown that they contribute to specific hallmarks of cancer. And given that circular RNAs are classified as a group based on their topology and not based on, say, function, people wondered how might they actually work. And so this shows a few different categories of circular RNA sort of function. The first one that was discovered and came into sort of public knowledge prominence was acting as a microRNA sponge. So an individual circular RNA encoded 70 copies that was antisense to a specific microRNA. And it was able to effectively sponge up that microRNA from acting on its microRNA targets. So now I've just explained that, essentially all eukaryotes contain these circular RNAs, and they can be quite abundant. There have been, maybe up to tens of thousands of different circular RNAs that have been identified. And sometimes they are, say, 10-fold the copy number as their linear RNA counterparts. Generally speaking, though, they are predominantly less than their linear RNA counterparts. Now, in this context of circular RNAs, people have taken patient samples with different types of diseases and found that circular RNAs are dysregulated. That means either the types of circular RNAs express change or the levels change. And now there's a lot of active research to understand, how do these circular RNAs actually contribute to the initiation and progression of these different types of diseases. Circular RNAs are actually also encoded in viruses. So the hepatitis delta virus was the, original circular RNA virus because it has a circular RNA genome. And this electron microscopy image was one of the first examples that circular RNAs actually existed in nature. Last year, 2 different groups found the Epstein-Barr virus as well as the Kaposi sarcoma-associated herpesviruses also encode circular RNA. So these are 2 DNA viruses that can lead to progression of cancer. So given that a healthy cell needs to maintain this vital balance between recognizing its own endogenous RNAs to prevent autoimmunity and recognize pathogenic RNAs to prevent infection, we had several different questions, essentially circling around how do cells do this for circular RNAs? So that was our first question. How do cells distinguish between self and non-self circular RNAs? Then we wondered, if they have this distinction and this ability to distinguish between self and non-self, can we harness that for specific therapies specifically related to cancer? We also wanted to understand what are the molecular mechanisms to sense foreign circular RNAs and self-circular RNAs. And finally, given all of these tens of thousands of circular RNAs, both virally and endogenously encoded, that become dysregulated in disease, what are the functions of these circular RNAs? And might we be able to target them for specific therapies? So to answer this first question, we needed to design a system that would model a foreign circular RNA. And to do that, we used the TD gene from T4 bacteriophage, which contains a group 1 intron. So this intron typically will autocatalytically splice itself into a lariat in order to join exon 1 to exon 2. We found that if you take this group 1 intron and permute the order, you take the 3' end and put it down upstream of a gene of interest, and the 5' end downstream of a gene of interest, whatever is in between these two pieces will autocatalytically splice itself into a circle during in vitro transcription without the addition of any other protein components. And so we can form this circular RNA. And we designed the sequence such that in the linear form, an internal ribosome entry site is downstream of our open reading frame. And during circularization, the IRES becomes upstream, enabling for translation of this open reading frame. This process during in vitro transcription also generates some linear RNA byproducts, which become substrates for an exonuclease called RNase R. So we were able to generate the circular RNA, which I'll call CircFOREIGN for the rest of the talk, and characterize it and purify it to homogeneity. So the first experiment that we did was we took the circular RNA and we transfected it into HeLa cells. And then we also chose a linear RNA with a 5' hydroxyl group because that should not be immune stimulatory, and also transfected it because we wanted to compare the sequence of the IRES and the GFP in between these two different topologies. We performed RNA sequencing where each dot is one of the genes that's found enriched in either CircRNA transfection compared to mock transfected cells, or linear RNA transfection compared to mock transfected. And the color of the dot represents the more red the dot is, the more enriched it is on the circular RNA transfected cells compared to the linear RNA transfected cells. So I've labeled some of the genes that we found up here, and you may notice they fall into a specific pathway. And indeed, when we performed gene ontology enrichment, we found that circular RNA transfected cells had strong induction of antiviral response and inflammatory response. And so given that cells respond and sense these foreign circular RNAs, what prevents self-circular RNAs from also being sensed and inducing this strong inflammatory response? And we hypothesized that potentially the RNA binding proteins that are associated with self-circular RNAs are distinct from the ones associated with foreign circular RNAs. And so to test this hypothesis, we performed an experiment using the comprehensive identification of RNA binding proteins by mass spectrometry, where we take live cells, we cross-link them, we use biotinylated silane oligos to enrich for our RNAs of interest. We can isolate that RNA with all of the proteins bound to it, and then identify the RNA binding proteins by mass spectrometry. So we took several different RNAs through this process. We took our foreign, circular, and linear RNAs through it, and then we also took a endogenous circular RNA through this process, but we made one change. So the ZK scan is a gene that encodes an endogenous circular RNA. And we wanted to test the biogenesis difference. And so we swapped out the endogenous ZK scan for our GFP IRES sequence. So right now, these CircFOREIGN and CircSELF sequences are identical. They're just made through two different processes, mimicking a foreign circular RNA and a self-circular RNA. And we found that a specific category of proteins came up as highly differentially expressed or binding to CircSELF and CircFOREIGN. And these are proteins associated with a RNA modification and 6-methyladenosine. So we find that there are M6A writers that install the modification. There are readers that recognize the modification, and there are erasers that remove this modification. And so here's the actual sort of catalytic cycle of the RNA modification. So given that potentially M6A is a molecular marker for self-circular RNAs compared to non-self-circular RNAs, we wondered maybe there's a differential modification of these different circular RNAs. And so to test this hypothesis, we used a method called RCLIP with M6A, where we use an antibody that recognizes the RNA modification. We can cross-link it so that the antibody is covalently attached to the RNAs that's modified. We can perform limited proteinase K digestion, leaving a scar of the protein attached to the RNA modification. This scar is large enough such that during reverse transcription, the polymerase will hit this scar and stop there, meaning that using this method with sequencing, we can have nucleotide-specific level of identification of exactly where the RNA modification exists. And so we took both the CircSELF and CircFOREIGN through this process, and we found that right after the splice junction, there's a significant peak of M6A density only on CircSELF compared to CircFOREIGN. And so we potentially wondered maybe CircSELF contains this RNA modification in order for the cells to sense it as self. And so then that means that we should be able to take this RNA modification, incorporate it into CircFOREIGN, and that should mask the foreignness of the circular RNA. And so when we try that experiment, that's exactly what we found. So if we take unmodified circular RNA, we get strong induction of this panel of innate immune genes. However, when we add in 1% M6A into the CircFOREIGN, where out of 100 A's, one of them is replaced with an M6A, we get about 50% reduction in the immunogenicity. So 1% is just about endogenous levels that we would find in the cell. And so this was really exciting to us. And then we wondered, you know, is there a way that given the cells can distinguish between self-circular RNAs and non-self-circular RNAs, might that be useful in some context? And so first we took cells in vitro and we transfected them with either linear RNA or circular RNA that's labeled with a fluorophore. Then we treated those cells and subjected them with a positive single-stranded RNA virus, the Venezuelan equine encephalitis virus, that's tagged with GFP, so we would be able to tell whether or not the cells were infected. And we find that indeed the cells that were first transfected with circular RNA were protected from subsequent viral infection. And we quantify the data here where the cells with circular RNA had significant lower levels of VEEV infection. We found, interestingly, that even the cells that did not themselves receive the circular RNA, but were in the same neighborhood or, you know, essentially well of the circular RNA, also were protected. So then we wondered, could we extend this to an in vivo model and see if we can get systemic protection of an animal? And so we designed an experiment where we wondered if we could use foreign circular RNAs as a vaccine adjuvant. And we used ovalbumin as a model antigen here. And so we immunized the mice twice, and then we collected T cells and probed the antibody titer for both of them. And here we see this is ova-specific CD8 plus T cells that both foreign circular RNA with a transfection reagent and without a transfection reagent was they were equally effective as our positive control poly-IC at inducing the same levels of CD8 plus T cell response. And we were really puzzled by the fact that, you know, whether we did not need an in vivo transfection reagent, and this is something that we're trying to understand mechanistically and molecularly as well. When we looked at the antibody response, we found that similarly, both circular RNAs with the in vivo transfection reagent as the one without also produced similar levels as their positive control poly-IC. So then we wondered, would circular RNA be a method as a vaccination? And could we use this to prevent overexpressing tumors? And so here we wanted to vaccinate the mice first, and then 2 weeks later, establish B16 overexpressing melanoma tumors and measure the tumors and image them because the cells are tagged with luciferase. So here are images of the mice treated with either PBS or CircFOREIGN. And we find that there's a dramatic difference in the rate of the tumor growth between the two different sets of mice. And this translates into a doubling of survival for the mice treated with CircFOREIGN compared with negative control PBS. So this is with unmodified CircFOREIGN, and we wondered, maybe there is some context where we want to be able to modulate and tune the level of immune response. And so we wondered, could we introduce M6A into our CircFOREIGN to change and adjust the level of induction? And indeed, that's what we see here, where again, in vivo, we model similar results that we saw in vitro, where 1% of M6A introduced in the foreign circular RNA decreases the immunogenicity by about 50%. So we're really excited by these different findings, and we're hoping to find some specific in vivo models to continue and probe CircRNA's therapeutic purposes. So we think that some of these experiments should go hand in hand with a deeper understanding of the molecular mechanism, because if we can understand that side, potentially we can also give insight into the therapies as well. And so we had found previously that RIG-I, which is a cytoplasmic nucleic acid sensor, is necessary for detecting the presence of foreign circular RNAs. Now, RIG-I typically is used to find viral RNAs with a 5' triphosphate. And so in the inactive state, the RIG-I has these card domains that fold up. However, in the presence of an agonist, RIG-I's card domains are now revealed, and then they're stabilized, either because there are other RIG-I filaments nearby, or in the presence of these unanchored polyubiquitin chains. And so we wondered, does circular RNA induce RIG-I in a similar way that these other viral RNAs would induce? And does the immune signaling sort of continue through this canonical pathway all the way to interferon production? So first we tested whether or not circular RNA itself was able to induce RIG-I filamentation. And we find that the answer is no. So here are electron microscopy images, and with our known agonist, a double-stranded RNA, we can see these nice white filaments, which is RIG-I forming these filaments. However, both in the presence of a 5' hydroxyl linear RNA, as well as with CircFOREIGN RNA, we were not able to induce these RIG-I filaments. Then we wondered, maybe if we add these unanchored polyubiquitin chains, that would be enough to stabilize the card domains. And so to probe that, we looked at whether or not we could activate RIG-I into a conformation that was able to trigger the next step, which is to trigger MAVS monomers to form these filaments. So now on these electron microscopy images, we would be looking for MAVS filamentation. So we did this in an in vitro system where we combined all of these different components, and then we modulated the concentration of the different RNA agonists and looked for presence of MAVS filamentation. And we find indeed, as we increase sort of circular RNA concentration, we get strong MAVS filamentation, but we don't with our negative control, our linear RNA. And then we can also modulate by the addition of M6A. And so we took these samples and we made EM grids. And you can see just by the number of filaments that unmodified CircFOREIGN strongly induced MAVS filamentation, and the addition of M6A RNA modification decreased that. And when we count the number of filaments and quantify them, we again observe this trend where just 1% of M6A is enough to decrease the immune response by about 50%. So After MAVS is formed, the next step is dimerization of IRF3. And when we probed whether or not circular RNA was able to make this induction, again, we saw IRF3 dimerization was forming, specifically in the presence of circular RNA. And so our model right now is that RIG-I is inactive until in the presence of unmodified circular RNA joined with these polyubiquitin chains, we can activate RIG-I to trigger MAVS filamentation, IRF3 dimerization, and subsequently interferon production. However, if there is RNA modification of M6A on the circular RNA, that inhibits this process. So RIG-I may bind, but it doesn't become into this active stage in order to trigger the rest of this immune signaling. So in the last minute, wanted to comment on, how can we study the different functions of circular RNAs? And could we break that up into some smaller questions to ask more specifically, what are circular RNAs doing in certain contexts? So right now, the field is really nascent and there are a lot of different unanswered questions. So it's really exciting to be in this area because there's so many things that we don't know. For example, how does a cell distinguish and choose when to make a linear RNA and when to make a circular RNA? There's been some bioinformatics studies showing that when the introns that flank a circular RNA are a little bit longer than usual and base pair, there more tends to be circular RNA produced, but that's not always the case. And so there's a decision in the nucleus during transcription and splicing to choose whether or not to forward splice to make linear RNA or back splice to make circular RNA. We're also interested in understanding the structures of the secondary structures of circular RNA. We hypothesize that they are quite different than their linear RNA counterparts because of the unique back splice junction, but exactly how they change and what functions they might have when they change are still unknown. We're interested in all the other chemical modifications. There's more than 160 that have been discovered. I just talked about one so far. And all the RNA binding proteins that associate with them and how that might contribute either to sensing of self, non-self, or other cellular processes. Circular RNAs endogenously are made in the nucleus, but they predominantly localize to the cytoplasm. Now, there's so many organelles in the cytoplasm that potentially if we can gain insight into organelle-specific localization of these circular RNAs, we might be able to understand what those circular RNAs doing there. And circular RNAs are also packaged in extracellular vesicles. So could this be one way, given their high stability, to be able to communicate between sort of just, disparate parts of the body? And what types of decisions are made to select which types of circular RNAs for these packaging and release? And how do cells actually, receive them and interpret the signal are still unknown. And finally, what are the circular RNAs on these organs and system levels? And sort of more importantly, what happens when this becomes dysregulated? I mentioned at the beginning that technology is really a key component to these types of investigations. And there's a strong need to develop new types of approaches to study circular RNAs, because for the most part, many RNA protocols start with selection of poly-A-tailed RNAs, and that automatically sort of eliminates all of the circular RNAs that would be present in the sample. And then can we take a deeper understanding on this area in order to apply circular RNAs to develop some useful therapies? So thank you very much for your attention. This is my small and hopefully growing group at Yale. And then I also did some of this work during my postdoc, where I was with Howard Chang and names of some of our collaborators and our funding sources. Thank you again.
Okay, thank you. Questions?
Is there a genetic way to prevent the formation of circular RNAs? And if so, has that gene been found to be altered in disease processes?
Yeah, so there is not one single way to prevent. There are a couple different ways to induce formation of. And there have been a couple proteins that have been found that if you put their recognition signal, you can get circularization. And then if you change the splicing machinery, you can also promote circular RNAs. So this is a really big question, and I think there's a lot of people sort of working hard to try and answer it. I don't know that there's going to be like a really clean answer.
So 2 questions. First, I assume these are virtually never translated, right? Because they eliminate the natural binding site.
Yeah, so that's also a big question. At the beginning, say, you know, 5, 6 years ago, people were doing a lot of different ribosome profiling, polysome fractionation, and they concluded that there was no translation. 2 years ago, 2 papers came out saying they found evidence of small proteins that could only come from translation across the backsplice junction. They haven't shown active translation on these circular RNAs, but they have sort of like a correlation.
And also, if you look, when we sequence tumors, I must say it seems to be an increasing phenomenon for some reason. It's not clear to me. But we see mutations in splice donors and splice acceptors quite frequently. Have you looked in those specific kinds of tumors for circular RNA?
Yeah, so many groups are taking just patient samples, just sequencing them as much as they can. The circular RNAs exist. They do change compared to sort of, you know, normal tissue. What their functions are and are the changes enough to drive something specific is still unknown. So I'm not sure.
But we could give you those tumors.
Great. That'd be fantastic.
Those genes.
Yeah. That'd be fantastic. I mean, I'm really interested in getting samples. We have pipelines that we work really hard to be able to find circular RNAs from sequencing. So I'd love to hear and chat more.
Yeah. You showed that circular RNAs can act as an adjuvant vaccination. Do linear RNAs not have that activity?
It's suppressed. Yeah, so we were using a 5' hydroxyl linear RNA, which we know is not supposed to be immune stimulatory. We've started doing some different types of experiments to see, you know, does the increased stability from circular RNAs yield additional benefits compared to these linear RNAs? Yeah.
Well, Grace, thank you. That's wonderful work. Terrific grand rounds from both
Thank you.
parts. Thank you. Really interesting. Certainly timely related to obviously Pico trade.
Yeah, right.